5.1 is on meiosis. Nothing lives forever, and therefore all living things must pass down their genes, which are their molecular instructions to their offspring. Heredity is the study of how this happens. Mitosis is an example of asexual reproduction, where one cell simply copies its DNA and splits into two clones. Multicellular life, in contrast, produce offspring that have unique combinations of genes or variation from two parents, which comes from sexual reproduction. Special cells called gametes or sex cells transmit a unique set of genes from each parent and then fuse to form a new organism with a brand new combination of genes. Meiosis is the type of cell division that shuffles the genes and chromosomes each parent passes down, producing 4 haploid cells, aka gametes, which have only one set of chromosomes each. When two haploid cells like a sperm and egg cell combine through fertilization, it forms a new diploid cell with 2 sets of chromosomes. In humans, meiosis produces sperm and egg cells with 23 chromosomes each. When the sperm and egg fuse, the future baby will have 23 homologous pairs of chromosomes, 46 in total, with never before seen combinations of genes, which means you are unique and special. 5.2 is about why everyone is genetically different. Meiosis produces 4 non-identical haploid gametes, so 2 divisions need to happen. The homologous pairs, or the pairs in which one chromosome was inherited from each parent, are split first during meiosis 1, and then the sister chromatids, or copies of each chromosome, are split during meiosis 2. Meiosis 1 begins with one diploid cell with duplicated chromosomes and ends with 2 haploid cells with duplicated chromosomes. Meiosis 2 thus ends with 4 haploid cells with unduplicated chromosomes, or basically just one set. Just like mitosis, both meiosis 1 and meiosis 2 have the same general phases. Meiosis ensures genetic variation through 2 main things. One is independent assortment, which basically means that homologous chromosomes are randomly divided between the 4 gametes so long as each gamete has at least one of each chromosome. This means that each gamete can get any random combination of the parental chromosomes. Second is crossing over, where homologous chromosomes basically swap genes with each other by touching, thereby increasing the number of combination of genes on each chromosome. These 2 factors make sure everybody's different from everybody else. 5.3 is on Mendelian genetics. Genetics is basically using heredity to analyze the inheritance of traits and make predictions. Gregor Mendel discovered that the inheritance of traits can be predicted using laws. He crossed pea plants in his garden, and it basically showed that some traits get masked behind others. Although Mendel didn't know what DNA was, he found out that these masked traits were produced by the inheritance of 2 recessive alleles, one from each parent. And thus, the trait that did the masking and came up more frequently in the offspring was the one that only required one allele to be expressed, which is what we call dominant. Alleles are simply versions of the same gene. The alleles an organism inherits are called its genotype, and the genotype determines an organism's phenotype, or its physical traits. Punnett squares use Mendel's laws to predict the outcome of a genetic cross, given the genotypes of the parent organisms. The ratio of offspring produced by 2 heterozygotes, or organisms with 2 different alleles at a gene locus, will be 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive, resulting in a phenotypic ratio of 3:1. 5.4 is all about the exceptions to the laws of inheritance that Mendel established with his experiments. Let's start from the top. Sometimes 2 alleles for a characteristic are both dominant. Other times, neither are. Sometimes one gene affects multiple phenotypes. Other times, multiple genes impact only one phenotype. Sometimes the inheritance of one gene at one locus alters the phenotype at another. Some genes aren't even part of the nuclear DNA, but are inherited through mitochondrial DNA. Sometimes genes in question are located on a sex chromosome, like X or Y. All of these break the rules of Mendelian genetics, but here's the big one. By doing test crosses for 2 different traits with fruit flies, Thomas Hunt Morgan found that some sets of genes are more likely to be inherited together than separately, leading to the discovery that some genes are located on the same chromosome, or linked. Basically, genes that are closer together on a chromosome are less likely to get flip-flopped during crossing over, and genes that are further apart tend to get swapped. This flies in the face of the law of segregation that Mendel wrote by saying that genes are inherited independently of one another. Topic 5.5 is on the environmental effects on phenotype. This whole unit has mostly been about the patterns in which an organism's genes impact its physical characteristics, or its phenotype. But obviously not everything about you is predetermined by your genes right when you're born, right? That would be kind of sad. But anyway, the environment plays a large role in phenotypic variations. Phenotype plasticity is the term that we use to describe when a phenotype is altered by the environment an organism lives in, even if it has the same genotype as the same type of organism in a different environment. In humans, things like skin color, height, weight, and intelligence are multifactorial, or all affected by both genetics and the environment. Here's some more obvious examples. The sex of reptiles when they're born is determined by the temperature at which their eggs are incubated. In orchids, the color of the petals is determined by the pH of the soil that the flowers are planted in. Topic 5.6 is on chromosomal inheritance. Earlier in this unit, we discussed how chromosomes are assorted independently during meiosis, and that each sex cell gets its own unique set of chromosomes, so that when it combines with another sex cell, the diploid zygote that arises from that fusion has a completely shuffled deck of genetic information. This is where genetic variation comes from. Studying how chromosomes are inherited has led us to a better understanding of genetic diseases, some of which result from a mutation in a gene located on a chromosome, or from the inheritance of too many or not enough chromosomes. Disorders like these have been better understood once we have determined their pattern of inheritance, like is the mutated allele dominant, recessive, autosomal, or sex-linked. Some disorders are caused by a non-disjunction of chromosomes during meiosis, where chromosomes do not properly separate from one another, leading to one gamete having too many chromosomes and another not having enough. Having an odd number of chromosomes like this is called aneuploidy, and it leads to disorders like Down syndrome or trisomy 21.